Hi everyone, my name is Ungbi Kwan from the Stein Monogastric Nutrition Laboratory at University of Illinois. Today, I'm glad to share with you some data related to branched-chain amino acid nutrition for growing pigs. And this is also part of my PhD dissertation work. Leucine, isoleucine, and valine, they are indispensable amino acid for swine. And they are categorized as the branched-chain amino acid because of the structural similarity of their side chain. For this reason, they have the interaction in their metabolism, for example, the antagonistic effects of excess leucine. Previous researchers indicated that if pigs consume excess leucine diets, they surprisingly can detect branched-chain amino acid imbalance within 1 hour of ingestion. And most studies indicated that excess leucine reduced their growth performance, especially average daily feed intake. And they concluded that the troublemaker in excess leucine diet was KIC. KIC is alpha-keto isocarproate, which is the metabolite of leucine. This compound can stimulate the enzymatic degradation of all three branched-chain amino acids, which means excess leucine can create the antagonism among the branched-chain amino acid. So previous research has been conducted to investigate excess leucine effects, and we concluded that excess leucine reduced growth performance and nitrogen retention. And we also confirmed excess leucine increased catabolism of all three branched-chain amino acids. Interestingly, we found one more thing. Excess leucine reduced serotonin concentration in the brain. Based on this serotonin results, we could explain why average daily feed intake was affected by excess leucine. Because serotonin is the neurotransmitter that is involved in feed intake regulation. Since tryptophan is the precursor of serotonin in the brain, we were thinking there must be something in between leucine and tryptophan. So we've conducted second study with leucine and tryptophan and concluded leucine had the negative effects on tryptophan uptake in the brain. And adding more tryptophan increased serotonin concentration in the brain, and it also increased average daily feed intake and average daily gain. But it couldn't fully overcome the negative effects of excess leucine. In addition to the negative effects on tryptophan utilization, if excess leucine reduced the efficiency of utilization of isoleucine and valine, which are the other two branched-chain amino acids, how we handle this problem. Therefore, in this study, we tested the hypothesis that adding more valine and isoleucine will overcome detrimental effects of excess leucine on nitrogen balance and branched-chain amino acid metabolism of growing pigs. So we utilized 144 growing barrels with initial body weight of 28.5 kilograms, and they were housed in metabolic crates and were assigned to 18 dietary treatments. We formulated the basal diet using less amount of corn and soybean meal and greater amount of wheat and barley, because this is the only way we could get the diet containing SID leucine level at the requirement. Otherwise, we will always overformulate diets above the SID leucine requirement. In order to investigate the effects of increasing levels of dietary leucine, isoleucine, and valine, we use crystalline L-leucine, L-isoleucine, and L-valine. Finally, we had 2 levels of leucine, 3 levels of isoleucine and valine respectively. One thing I want to point out, the dietary concentration I put here with orange colors, they are the requirement of each branched-chain amino acid from NRC 2012. So 3 levels of each isoleucine and valine represent below the requirement, at the requirement, and above the requirement. Therefore, we had 18 dietary treatments with different SID branched-chain amino acid levels. After 5 days of adaptation period, total amount of urine and feces were collected for 5 days according to the marker-to-marker procedure. For sample collection, all pigs were euthanized on day 13. Blood samples were collected on both day 0 and day 13. Tissue samples of liver and muscle were collected right after euthanization. Before we jump into the results, Since we couldn't find any three-way interaction for all response criteria from this 2x3x3 factorial treatment arrangement, I'd like to show you the data based on two-way interaction and main effects. Let's see the nitrogen valence data first. For nitrogen retention, we only found leucine effect. As we added more leucine, nitrogen retention was decreased. For biological value of protein, we had exactly same trend here. As we added more leucine, biological value of protein was decreased, indicating that excess leucine has negative effects on nitrogen balance in growing pigs. How about plasma urea nitrogen? There was a similar trend as we saw in nitrogen balance data, but we also found the valine effect. As we added more leucine, plasma urea nitrogen was increased. However, as we added more valine, plasma urea nitrogen was decreased. Therefore, adding more valine is beneficial in terms of efficiency of amino acid utilization. And this is the data of brain-chain amino acid concentrations in skeletal muscle. For all three brain-chain amino acid, we only found leucine effects. As we added more leucine, concentrations of all three brain-chain amino acid in skeletal muscle were decreased. How about brain-chain amino acid concentration in the liver? We found only leucine effects here as well. However, unlike skeletal muscle, as we added more leucine, concentrations of all three brain-chain amino acid in the liver were increased. Based on the data from these two different tissues, we can conclude that excess leucine could change the brain-chain amino acid composition and their metabolism in the body. Now, let's see the plasma-free brain-chain amino acid data. For isoleucine, we found the interaction between leucine and isoleucine, leucine effect, and isoleucine effect. As we added more leucine, plasma-free isoleucine decreased. However, as we added more isoleucine, plasma-free isoleucine increased. According to the interaction, the increase is greater if we have lower leucine in the diet. How about valine? Exactly the same trend here as well. We found the interaction between leucine and valine, leucine effect, and valine effect. Like I mentioned before, these plasma-free isoleucine and valine data indicate that excess leucine has clear negative effects on availability of isoleucine and valine for protein synthesis. For leucine, it's pretty obvious we found only leucine effect. The more leucine we have in the diet, the more leucine in the plasma. Now, let's see the brain-chain alpha-keto acid data. We analyzed three different brain-chain alpha-keto acids from the metabolism of brain-chain amino acid in the plasma. For KMV, which is the metabolite of isoleucine, we found interaction between leucine and isoleucine, leucine effect, and isoleucine effect. As we added more leucine, plasma KMV decreased. However, as we added more isoleucine, plasma KMV increased. The increase is greater if we have lower leucine in the diet. For KIV, which is the metabolite of valine, we had a similar data here as well. As we added more leucine, plasma KIV decreased. However, as we added more valine, plasma KIV increased. According to the interaction, increase is greater if we have lower leucine in the diet. Notice that these results for KMV and KIV have similar trend what we saw in the plasma-free isoleucine and valine. However, for KIC, which is the metabolite of leucine, we couldn't find any interaction, but we find leucine effect and valine effect at the same time. It was surprising because we were only expecting increased KIC concentration in excess leucine, but we didn't expect the valine effect. As we added more valine, plasma KIC was clearly decreased. This indicates that adding more valine could inhibit the stimulating effects of KIC on brain-chain amino acid catabolizing enzymes by reducing KIC concentration in the blood. In conclusion, we confirmed the negative effects of excess dietary leucine in growing pigs in terms of nitrogen retention, efficiency of amino acid utilization, and availability of the other two branched chain amino acids for protein synthesis. However, valine supplementation can partly overcome the negative effects of excess leucine in terms of efficiency of amino acid utilization, and it might reduce the catabolism of branched chain amino acid by reducing KIC concentration in plasma. I would like to thank the sponsor, Ajinomoto, for financial support, and I want to acknowledge all the members in the Stein Monogastric Nutrition Lab. If you want to learn more about our research, please visit our website. Thanks for your attention.